Ever wish you could like hold the actual blueprint to a brain
Oh,
in your
yeah.
hand?
Yeah.
Well, get this. Scientists have gotten about as close as you can get with a fruit fly.
Right.
Yeah,
It's called FlyWire.
Okay.
And it's the most complete map of neural connections ever created for any brain.
Wow.
We're talking 139,255 neurons and 15.1 million connections. A 3D wiring diagram.
Wow.
More intricate than anything we've seen before.
15 million connections.
Yeah.
That's mind-boggling.
It's a lot.
But why a fly?
Right.
What can we learn from something that, let's face it, most people swat away without a second thought?
Yeah, well, it's easy to underestimate the humble fly, but their brains,
Sure.
despite their size, share fundamental organizing principles with our own. So by decoding the fly brain, we're actually uncovering secrets about how brains in general work.
So it's like using a simpler model
Yeah.
to understand a much more complex system.
Exactly.
Okay.
FlyWire isn't just a map, though.
Okay.
Think of it as a parts list
Okay.
for a brain,
Right.
meticulously categorized.
Oh, wow.
Each neuron is tagged based on its location, its shape, even what it connects to. This is crucial when you're dealing with this level of complexity.
Yeah, I imagine. So it's one thing to see all these connections, but it's the organization, the cataloging
Right.
that makes FlyWire truly revolutionary.
Precisely.
Okay.
This system allows researchers to zero in on specific types of neurons and their connections.
Okay.
For example, they can isolate the neural pathways for smell, or those associated with memory, even those that control flight.
Wow. So it's like having a Google Maps for the fry brain,
Yeah.
but with filters for every type of neuron and connection.
That's a great analogy.
Wow. You can really zoom in on the details.
But FlyWire goes even further.
Okay.
The researchers didn't just map the connections, they analyzed their strength.
Okay.
They discovered that connections with more than 10 synapses, the junctions between neurons,
Right.
are much more likely to be functionally significant, not just random noise.
So it's about finding the neural highways,
Exact.
the connections that are really important for the brain to function.
Exactly.
And what's fascinating is that these strong connections are remarkably consistent across different flies.
That's right.
So you're telling me that even though each fly is unique,
Yep.
there's a fundamental blueprint for their brain.
There is.
That's mind-blowing. How does that even work?
Well, that's where things get even more interesting.
Okay. Yeah.
To understand this consistency, the researchers had to differentiate
Okay.
between what was simply background noise in the data
Sure.
and true biological variation
Okay.
between flies.
So like trying to pick out a single voice in a crowded room.
Yeah.
How did they manage to do that?
They analyzed data from multiple fly brains, allowing them to identify the consistent patterns,
Okay.
the true signals, amidst the noise of individual variation.
So by comparing multiple brains, they were able to kind of filter out the static.
Yeah.
What emerged from that analysis?
So one of the things they focused on was a region called the mushroom body,
Okay.
which is essential for learning and memory in flies.
The mushroom body.
Yeah.
Okay. Now that's just cool sounding.
It's a great name.
Is it shaped like a mushroom?
It really is.
Get out.
And here's where the findings get even more intriguing.
Okay.
When they compared the FlyWire data to an earlier project that mapped part of a fly brain, the hemibrain,
Okay.
they found a significant difference in the number of Kenyan cells,
Okay.
the main type of neuron in the mushroom body.
So one brain had a lot more Kenyan cells than the other? Does that mean something went wrong?
Not at all.
Okay.
Remember, there's natural variation between individuals,
Right.
just like there is between humans.
Okay.
But what's amazing is that despite this difference in cell number, the overall organization and function of the mushroom body remain remarkably consistent.
Hold on. So you're saying the brain can adjust its wiring based on how many cells it has?
It seems
Like having a team with fewer players, but they still cover the entire field.
Exactly.
Wow.
They discovered that if there are more Kenyan cells,
Okay.
each one receives fewer inputs from other neurons.
Oh, interesting.
It's a remarkable adaptation strategy.
So the brain can adjust on the fly.
Literally.
Literally
Right.
to optimize how it's wired.
Yeah.
This is making me question everything I thought I knew about brains.
And this leads us to one of the biggest challenges in neuroscience.
Okay.
Defining what constitutes a cell type in the brain.
Because if one fly has more or fewer of a type of cell, how do we even know it's the same type across individuals?
That's exactly right.
Right.
It's like trying to categorize all the different types of trees in a forest.
Okay.
You have pines, oaks, that's easy. But there are so many subtle variations.
Right.
Where do you draw the line?
So how do you tell if a difference is significant or just a slight variation within a type?
Well, that's where the power of having multiple connectomes comes in.
Okay.
By comparing data from different fly brains.
Okay.
Researchers could identify cell types that were consistently distinct
Okay.
across individuals, regardless of minor variations.
Okay, so they're looking for the neuron equivalent of species.
Yeah.
Distinct enough to be their own category.
Exactly.
Did this analysis upend any previous assumptions from the earlier hemibrain project?
It did.
Really?
Some previously proposed cell types turned out to be less clear-cut than initially thought. This highlights how important it is to have these massive data sets for comparison and refinement.
So FlyWire isn't just giving us a bigger picture, it's making the picture sharper.
I like that.
And this all goes back to your earlier point about the fly brain being a model for understanding brains in general.
Absolutely.
Yeah.
The insights from FlyWire have profound implications, not just for understanding how brains develop and function, but also for potential medical breakthroughs.
Now we're talking...
Yeah.
You mean example.
Okay.
How could this help with human diseases?
Take Parkinson's disease, for example.
Okay.
It's caused by the loss of specific neurons.
Right.
If we can understand the different cell types involved and how they're connected, we might be able to develop more targeted therapies.
So instead of treating the symptoms, you could potentially target the specific circuits that are malfunctioning.
Exactly. But beyond the potential medical applications, FlyWire raises some really profound questions about how brains, these incredibly complex structures, are even possible in the first place.
All right. You've got me hooked.
Yeah.
What are these profound questions?
Well, think about it.
Okay.
You have billions of neurons in your brain.
Yeah.
Trillions of connections.
A lot.
But you also have billions of letters in your DNA, your genetic code.
Right.
The question is there enough information in your DNA to specify every single connection in your brain?
That's a lot of connections to cram into our genetic code.
It is.
I'm going to guess the answer is no.
You're right.
Okay.
It's what's known as the genomic bottleneck.
Okay.
The idea that the brain's wiring diagram is simply too complex to be entirely encoded in our genes.
Okay, so if our genes aren't telling our brains exactly how to wire up what is, how does this incredibly intricate organ come together?
That is the million-dollar question in neuroscience.
Yeah.
One possibility is that our genes lay down a basic framework, the major highways, and then experience, the environment, shapes the finer details of the connections.
So it's like our genes provide the blueprint for a house.
Right.
But the lived experience, the memories made within its walls, that's what shapes the final structure.
That's a great way to think about it. And this is where the power of connectomics, especially projects like FlyWire, truly shines. By comparing the connectomes of flies raised in different environments, or even flies with specific genetic mutations, we can begin to untangle this complex interplay of nature and nurture.
We could see which connections are hardwired, determined by our genes, and which ones are more malleable, shaped by experience.
Exactly.
That's amazing.
It is. But there's another crucial question we haven't addressed yet.
Okay.
Remember we talked about variation between individuals.
Right.
How do we know this one fly brain, meticulously mapped in FlyWire, is representative of flies in general?
Yeah, that's a good point.
What if we just captured a snowflake, something totally unique?
Right. Because if every fly brain is wildly different, then what we learned from FlyWire might not apply broadly.
Exactly. But here's the good news. The researchers found a remarkable degree of similarity, of stereotypy, across the fly brains they studied.
So even though each fly has its own unique experiences, there's a fundamental blueprint, a shared structure to how their brains are wired.
Precisely.
Okay.
And this consistency is especially true for those strong connections. The neural highways we discussed earlier, they're remarkably similar from fly to fly.
Okay, that makes sense. Those essential pathways probably don't change much. What about the weaker connections, the back roads? Are they all over the map?
Even there's a surprising level of consistency.
Really?
Especially when you look at what's called normalized connection strength.
Normalized connection strength. Break that down for me.
So it's not just about the absolute number of connections, but the proportion of input a neuron receives from its various partners.
So it's like looking at the balance of influence.
Exactly.
The ratio of input rather than just the raw numbers.
Exactly.
Right.
And this normalized connection strength turns out to be remarkably consistent across flies, even when there are variations in the number of synapses involved.
Wow. So even with individual variation, there's a deep underlying order.
Right.
A set of rules governing how these brains are wired.
Precisely.
Wow.
And that means that FlyWire isn't just a snapshot of one fly brain.
Okay.
It's a window into the fundamental wiring principles of an entire species.
That is mind-blowing.
Yeah.
So where do we go from here?
Right.
What's the next frontier in connectomics?
Oh, gosh, the possibilities are kind of endless.
Yeah.
Imagine comparing connectomes across different species. Okay. Seeing how brains have evolved different wiring solutions to solve different problems. Or studying how connectomes change across the lifespan, from infancy to old age.
We could unlock the secrets of development, of aging, of consciousness itself.
Yeah.
This really feels like a turning point in our understanding of the brain.
It does, doesn't it?
Yeah.
And it all started with a humble fly.
Right.
It just goes to show sometimes the biggest discoveries come from the smallest creatures.
This deep dive has been incredible.
It has.
From the intricate world of fly brains to the fundamental questions about what makes us who we are.
Yeah.
FlyWire is really pushing the boundaries of what we thought was possible.
It certainly is.
And it really highlights something essential about science.
What's
a willingness to look closely at the world around us, even the seemingly insignificant, can lead to profound insights.
I agree.
Absolutely. Well, on that note, I think it's time for us to wrap up this deep dive.
Okay.
Until next time, keep those synapses firing. Okay, get ready, because today we're diving deep, and I mean deep, into the world of root fly brains. You heard that right. We're taking on this fascinating research paper, whole brain annotation and multi-connectome cell typing of Drosophila. Before you hit that stop button thinking, fruit flies? Seriously? Oh, trust me on this one. It's way cooler than it sounds. We're talking about scientists creating a complete detailed map of a fruit fly's brain, a connectome actually. And get this, they're using it to unlock the secrets of different neuron types, those tiny brain cells that make everything tick. Expert speaker, you know how much I love a good brain teaser, so tell me, why should we, with our big human brains, care about what's going on inside the head of a fly?
Well, what's fascinating here is that size isn't everything. The fruit fly brain, despite being tiny, is incredibly intricate. This research uses a connectome, which you can think of like a massive wiring diagram showing how each neuron connects with the others. It's like trying to map out every single wire in a giant computer network. But this one runs on sugar and helps a fly decide where to land next. The FlyWire connectome they use here is the most complex one we've ever created. We're talking huge amounts of data. And get this, they didn't just use one fly brain, they used 3.
3 brains.
Okay.
Now you're just messing with me. Seriously, how do you even begin to make sense of that much information? It's like finding Waldo in a stadium full of Waldos.
Yeah, you're not wrong. It's a mountain of data.
Yeah.
To organize it, they created a system. Think of it like organizing your computer files. You've got your main folders, then subfolders, and finally individual files.
Okay, so far so good. I can handle folders.
Great. So first they organized the brain by region based on the actual structure. Then they looked at where each neuron came from during the fly's development, its developmental origins, we call it. Finally, they even factored in the neuron's shapes.
So shape, origin story, and location in the brain, it's like giving each neuron its own ID card.
Exactly. And here's where it gets really cool. They focused on groups of neurons called hemilineages. These are neurons with a shared family history, kind of like siblings.
Okay, so neurons with shared DNA tend to hang out in the same circles.
Often, yes. And they often have similar jobs within the brain, too. It means there's a really impressive level of organization to how a fly's brain develops. It's precise. We're talking the number of neurons in a fly's central brain differing by only 0.1% between the left and right sides.
Wow, that's incredible. Talk about consistency. But how do we know this isn't just one weird, super organized fly? Are all fly brains wired this consistently?
That's the million-dollar question, right? It's something scientists call the snowflake problem. The worry that each brain, like a snowflake, is unique. To figure this out, they compared the wiring diagrams from all 3 fly brains. And guess what? The strong connections, The ones with lots of synapses, those little points of communication, were practically identical across the board.
So like a city, there's a main grid of highways and avenues that everyone uses.
Exactly. But here's the kicker. The weaker connections, the smaller roads, if you will, those showed a lot more variation between individual fly brains. It suggests that while there's a basic blueprint for a fly brain, there's room for individual flair too.
So it's like you and I both have the same basic skeleton, but the way our muscles and tendons attach might be slightly different, giving us our own unique ways of moving.
I love that. It explains why some flies are better at dodging my fly swatter than others, I guess.
That's a great analogy, and it has major implications for how we think about brains in general, not just those of flies. Individual variations might be way more common than we previously thought.
Yeah.
But speaking of those individual differences, remember how we talked about how these researchers were initially thrown off by some missing neuron types? Oh, right. It's like they went to check out a book from the library, but someone had moved it to a different shelf. So what happened there?
Well, their initial goal was to confirm neuron types that had been identified in a previous study, which used a different data set called hemibrain. But when they compared notes, they found that about a third of the hemibrain neuron types weren't showing up in the FlyWire data.
Wait, a whole third? That's a lot of missing neurons. Were they just not there? Did the FlyWire flies skip a few steps in brain development?
It was a head-scratcher, for sure. Turns out it wasn't that those neurons were missing entirely. It was more that the way they were defining neuron type was a bit too rigid. They needed a new system, one that accounted for all the subtle variations between individual brains.
So, like, instead of organizing books by the color of their covers, they needed to start using the Dewey Decimal System.
Exactly. They realized that just looking at a neuron's shape, which is what they'd been doing, wasn't enough. It's like assuming that all books with red covers are romance novels. Sometimes you get a cookbook in there, too.
Okay, so if the old way of IDing neuron types wasn't cutting it, what's the new and improved method?
They shifted their focus to connectivity, looking at how neurons are actually wired together. They realized that even if two neurons look a bit different, if they're making similar connections with other neurons, they probably belong to the same club, functionally speaking.
So it's about who you know, not what you look like. Makes sense. My mom always told me it's about personality, not appearances.
Exactly. And this new way of defining cell types is way more powerful. By applying it to the FlyWire data, they were able to pinpoint over 8,000 different cell types in that tiny fly brain.
8,000. That's like more cell types than there are people in my hometown. And that's all inside a brain the size of a poppy seed. Mind officially blown.
It's pretty incredible, right? And here's the kicker. Those 8,000 cell types aren't just theoretical. They've been cross-checked using multiple brains, which makes them much more reliable than anything we've had before.
So this isn't just some random fly brain census. We're talking about a highly accurate, thoroughly vetted head count of neuron types. But hold on, we've been talking a lot about neuron types, but what about the bigger picture? What did all this tell us about how brains are actually organized?
That's the beauty of this research. It's not just about counting neurons. Remember those strong and weak connections we talked about earlier? Well, they turned out to be key to understanding how brains balance consistency with flexibility.
Okay, I'm intrigued. Tell me more about this balancing act.
Think of it like this. The strong connections, those busy highways, are consistent across brains. That makes sense? They're probably in charge of essential functions, things you don't want to mess with too much.
Right, like how breathing is pretty much the same for all of us. Don't need a ton of variation there.
Exactly. But those weaker connections, the side streets, those showed a lot more variation between individual fly brains. This suggests they're involved in things like personality, learning, maybe even those fancy flight maneuvers some flies have.
So we've got the essentials, and then there's room for personalization. I like where this is going.
Precisely. And this isn't just true for flies. It challenges us to rethink how we define cell types in more complex brains, maybe even our own.
You're saying our brains might be way more customized than we ever imagined. Okay, that's officially going on my list of fun facts to bust out at parties.
Oh, absolutely. And this research gives us the tools to start exploring that individual variation in much greater detail. It's like we've had this incredible map of the brain sitting right in front of us, but now we finally have the key to unlock all of its secrets.
Speaking of secrets, we touched on something earlier that really caught my attention. The mushroom bodies and those Kenyan cells that are involved in memory. You mentioned they were a bit unpredictable.
Ah, yes. The Kenyan cells, they're like the rock stars of the fly brain, always the center of attention.
Well, they don't call it the fly mind for nothing. Okay, what's the scoop with these Kenyan cells?
They're found in a part of the fly brain called the mushroom body, and they're vital for learning and memory, particularly when it comes to smells. And what's interesting is that the number of Kenyan cells varied quite a bit between the FlyWire brains and the hemibrain data. We're talking a 30% difference.
Whoa, 30%? That's a bigger difference than my last online shopping spree versus what I actually told my partner I spent.
Quite a significant difference, yes. And it wasn't just the overall number either. One specific type of Kenyan cell, called KCGM, was way more abundant in the FlyWire brains compared to the hemibrain data.
KCGM sounds like a trendy new energy drink. So what makes this KCGM so special? And why were there so many more in the FlyWire flies?
That's the million-dollar question. It seems even in the highly organized world of the fly brain, there's room for individual quirks. The researchers have a few theories, though. Remember how we talked about the importance of a neuron's connections?
Oh, right. Like that whole it's not what you look like, it's who you know thing.
Yeah, exactly. Well, the KCGM neurons in the FlyWire brains, despite being more numerous, were actually getting input from a smaller number of odor channels than their counterparts in the hemibrain data.
Okay, hold up. Odor channels. We're talking about brains here, not cable TV.
Fair enough. Think of it like this. Your nose is picking up tons of different smells all the time, right? Each smell has its own unique chemical signature.
Sure, like freshly baked cookies versus my dog after a bath.
Exactly. Well, in the fly brain, each odor channel carries information about a specific type of smell. And those KCGM neurons we're talking about, they're like the brain's smell experts, and they get their information from these odor channels.
So more KCGM neurons, but each one is getting information from fewer odor channels. It's like having a bigger team, but each member has a more specialized role.
That's a great way to put it. It seems the brain likes to keep things balanced. Too many odor channels, and the poor fly might get overwhelmed with all the sensory input, but too few, and they might miss out on those crucial smells, like overripe bananas, a sure sign of a delicious meal if you're a fruit fly.
So even with all these individual differences in neuron numbers, the brain is finding ways to compensate and keep things humming along smoothly. It's like some kind of intricate neural ballet up there. But I have to ask, did they find similar patterns in other types of Kenyan cells? Or were the KCGM neurons just being divas?
Oh, this wasn't just a solo act. The researchers found similar patterns of compensation in other Kenyan cell types, too. It seems like the brain has a whole toolbox of strategies for dealing with variations in neuron number, ensuring everything works in sync. It's a beautiful example of adaptability.
It's like no matter what kind of hand you're dealt in terms of neurons, your brain is a master at playing the game.
Oh, yeah.
I'm starting to think we underestimate these little guys.
I agree. And it speaks to the incredible power of this multi-connectome approach. By comparing data from multiple brains, we're getting a much richer, more nuanced understanding of how brains are wired and how they adapt. It's a game changer.
So where do we go from here? This whole multi-connectome thing feels like a pretty big deal.
It absolutely is. This research is just the tip of the iceberg. Think about it. If we can uncover these kinds of insights from a brain the size of a pinhead, imagine what we can learn from more complex brains.
Okay, my mind is already racing. But before we start building connectomes of the entire animal kingdom, let's zoom back in for a second. We've covered a lot of ground here. Can you give us a quick recap of the biggest takeaways from this research?
Absolutely. First, size isn't everything. The humble fruit fly brain is proving to be a treasure trove of information about brains in general. Second, while there's a basic blueprint for how brains are wired, there's also a surprising amount of individual variation, which might be key to understanding things like personality and learning.
Right, like those quirky Kenyan cells.
Exactly. And finally, this multi-connectome approach, where we compare wiring diagrams from multiple brains, is crucial for getting a more complete and accurate picture of how brains are organized. It's like comparing different editions of the same book. You start to see which parts are essential and which parts have a little wiggle room.
It's like we're finally starting to understand the language of the brain, but instead of words, we're deciphering connections.
Beautifully put. It's an incredibly exciting time to be studying brains.
It really is. It's like cracking a code, but this code contains the secrets to how we think, learn, and perceive the world. It's mind-blowing. But before we get too lost in the philosophical weeds here, I have a more practical question. How is all of this even possible? I mean, we're talking about mapping millions, maybe billions of connections in a brain that's barely visible to the naked eye.
It's a massive undertaking for sure. It requires some seriously cutting-edge technology and a whole lot of brain power, pun intended.
I appreciate that.
The first step is to image the brain using a technique called electron microscopy.
Electron microscopy. Okay, that rings a bell. Isn't how we get those amazing close-ups of atoms?
You got it. It uses beams of electrons instead of light to create images. And it lets us see things in incredible detail. We're talking about being able to zoom in on individual synapses, those tiny junctions between neurons.
Okay, so we've got these super detailed snapshots of the brain, but how do we go from a bunch of static images to a dynamic three-dimensional wiring diagram? That's where FlyWire comes in, right?
Exactly. FlyWire is more than just a data set. It's a powerful online platform that allows scientists and even citizen scientists from around the world to collaborate on reconstructing neurons and mapping connections.
Wait, citizen scientists?
Yeah.
Like regular folks who just happen to be brain enthusiasts.
You got it. It's like a giant online jigsaw puzzle where each piece represents a tiny segment of a neuron. Anyone can log in, choose a piece, and start tracing its path.
That's so cool. So it's not just a few experts in a lab somewhere. It's a global community working together to solve this brain puzzle.
Exactly. And it's not just about tracing the neurons either. FlyWire lets us identify cell types, label brain regions, and ultimately create this incredible multi-layered map of the fly brain.
It sounds like FlyWire is revolutionizing the field of connectomics, making it more accessible and collaborative.
Absolutely. And this is just the beginning. As imaging technology and computational tools continue to improve, we can expect even larger and more complex connectomes in the future.
It makes you wonder, what will we be able to uncover when we can map 1 million brains, or even 1 billion?
The possibilities are, well, pretty much endless. This research on fruit flies is just a glimpse into the vast landscape of the brain. But it's showing us that even the smallest brains can hold profound clues about how brains evolve, how they function, and how they give rise to complex behaviors.
Well, expert speaker, this has been an incredible journey. We've covered everything from neuron ID cards to odor channels to the power of citizen science. I have to say, when we started, I wasn't sure what to expect from a deep dive into a fly brain, but it's blown my mind wide open.
I'm glad to hear that. It's a reminder that amazing things can come in small packages and that sometimes the most unexpected creatures can reveal profound truths about ourselves and the world around us.
It definitely makes you appreciate the incredible complexity hidden inside every living thing. Well, on that note, we've reached the end of our deep dive into the world of fruit fly brains and the groundbreaking research happening in the field of connectomics. A huge thank you to expert speaker for guiding us through this fascinating landscape. And to our listeners, thank you for joining us. We'll catch you next time on another deep dive. Hey everyone, ready for another deep dive? Today, things are going to get a little buzzy.
Yeah.
We're talking fly brains. Yeah, you heard me right.
Now, before you click away thinking, fly brains, really? Trust me, this is cool stuff. We're diving into a paper called Whole Brain Annotation and Multi-Connectome Cell Typing of Drosophila.
Catch you right. Don't worry, we'll break it down.
Basically, we're talking about a map, like a super detailed map, but for a fly's brain.
So like showing all the little roads and highways inside their tiny little heads?
Exactly. Except instead of roads, it's neurons, and instead of cars, it's electrical signals zipping around.
Okay, so this brain map. What makes it so special? I mean, haven't scientists mapped brains before?
They have, but this one, called FlyWire, it's the most complex one ever made. It maps out over 139,000 neurons and millions of connections between them. To put that into perspective, that's way bigger than the previous record holder, the hemibrain, which only looked at, well, half a fly brain.
Wow, that's a lot of neurons. More than I can even imagine. So how do scientists even begin to make sense of all that information?
That's where annotation comes in. Imagine looking at a giant tangled mess of wires, except you have no idea what any of them do or where they go. Annotation is like labeling all those wires, figuring out which ones connect to what.
So like giving each neuron a name tag and a job description.
Pretty much. This paper, they created a whole system for organizing these neurons based on where they are in the brain, what they do, that sort of thing.
So like a filing system for brain cells.
Yeah, exactly. And get this, one of the coolest things they found was this hemilineage atlas. It's like a family tree, but for neurons. They traced each neuron back to its parent cell.
A family reunion, but for brain cells. That's wild.
And you know what's even wilder? They found that the number of neurons in each of these neuron families was incredibly consistent between the left and right sides of the brain. It's like there's a stripped blueprint that says each family gets this many neurons, no more, no less.
Wow. That's incredible. So we're talking about this incredible level of organization, this intricate blueprint for a brain that's, what, the size of a poppy seed. It's mind-blowing. Okay, so we've got this amazing map of the fly brain.
Right.
But, like, a map's only useful if you know where you want to go, right?
Exactly. It's not just about the where, it's about the how.
So how do we go from this map of neurons to actually figuring out how a fly thinks, how it sees, how it does, fly stuff?
Well, it comes down to the connections, not just which neurons are connected, but how strongly they're connected.
Okay.
And that's where this idea of edge weight comes in.
Edge weight. Sounds like something you'd measure at the gym.
Well, it's kind of like that, actually.
Yeah.
Imagine you've got two friends, right? One of them, you text back and forth all day, every day. The other one, maybe you exchange a message once a week.
Yeah.
That difference, how often you're communicating, that tells you something about the strength of the connection. Yeah.
Okay, I'm following you.
So in the brain, a strong connection, a high edge weight, means there are tons of synapses.
Like those little bridges between neurons?
Exactly. Tons of synapses firing between those two neurons. And the researchers, they found something really interesting about these high edge weight connections.
What's that?
They were much more likely to be the same across different flies.
Oh, interesting. So, even though each fly's brain is a little bit different,
Right.
there are still these core communication lines.
Exactly. These core communication highways that are basically the same.
Huh. Like a biological internet.
Yeah. And from there, they came up with these two kind of rules of thumb for understanding the data.
Okay, I like rules of thumb.
First rule, if it's got more than 10 synapses, it's probably important. That's a real connection, not just some random thing.
Right, you wouldn't build a whole highway for just one car to drive on.
Exactly. And second rule, if the connection strength is only slightly different, like less than 30%, it might not actually be a real difference.
Oh, wait.
It might just be, slight variations in how they map to the connectome, not actual differences in the brains themselves.
So it's like filtering out the noise to find the real signal?
Yeah, exactly. Focus on the strong connections, the consistent ones, because those are the ones that really matter. Now, that's all well and good, but they wanted to put this whole edge weight thing to the test, right? So they decided to compare their fancy new FlyWire connectome,
The super detailed, right?
with the older hemibrain data.
Which was only half a brain, right?
Exactly. Now, you'd think same species, different flies, sure, but the brain should be basically the same, right? You wouldn't expect to find totally different parts in a Honda Civic versus, a Honda Accord.
Right. They'd have the same basic blueprint.
Well, that's not quite what they found. Over 30% of the cell types they had identified in the hemibrain data, completely missing in FlyWire.
Whoa, wait, missing. Like, poof, gone.
That's what they thought at first.
Okay, so we've got these missing cell types. Is this we haven't found them yet missing? Or they genuinely don't exist in these flies missing?
That's the million-dollar question. It could be that these cell types are really, truly absent in some flies,
Wow.
which would point to a crazy amount of individual variation, right?
Well,
Or maybe they're there, but we just need a new way to see them. Maybe our tools aren't quite good enough yet.
so it's either lost cities in the fly brain, or we need a stronger magnifying glass.
Exactly. And get this. Remember we talked about the mushroom body, that part of the brain that's super important for learning and memory?
Yeah, the brainy stuff.
Well, in FlyWire,
Okay.
the flies had something like 30% more Kenyan cells.
Wait, hold on. 30%?
Yeah.
That's not just a rounding error. That's a whole extra chunk of brain cells.
I know, right?
Kenyan cells, those are those neurons in the mushroom body.
Right, exactly.
Wow. Okay, so now I'm really questioning everything I thought I knew about brains. If they can be that different even within the same species.
That's what they started thinking, too. It really makes you step back and say,
Yeah.
what even is a cell type,
Right. If it can vary that much.
Exactly. So they thought maybe instead of just looking at individual brains and saying, oh, this cell is different because of this one little quirk, what if we look for similarities across different brains?
Oh,
Find the things that are consistent.
these cells are all part of the same family.
Yes.
Yeah.
Even if they have their own little quirks, they share these core traits.
So zoom out to see the bigger picture.
Exactly. And that's
Yeah.
exactly what they did. They called this the multi-connectome approach. Basically, they took 3 fly brain hemispheres,
Okay.
2 from FlyWire and 1 from hemibrain.
Okay.
And they looked for groups of neurons that were more similar across those 3 brains.
So more similar to each other than they were to other neurons in their own brains.
Yes.
Yeah.
Can we identify the family resemblance even with the individual quirks?
Wow. That's really clever. So did it work?
It did.
Yeah.
Not only did they validate a ton of existing cell types, but they also found thousands of potential new ones.
Thousands.
Thousands. They opened up this whole new world of cell type diversity that we didn't even know existed before.
Wow. We started with a fly brain map, stumbled upon some missing cell types, and ended up revolutionizing how we classify neurons. This is why I love deep dives.
Me too. And, it really makes you think about how much we still don't know, even about something as, well, seemingly simple as a fly brain.
It's true. When you think about it, there are more neurons in a fly brain than there are stars in the Milky Way galaxy.
Yeah.
And if their brains are that complex, just imagine what's going on in ours. Well, folks, another deep dive in the books. Thanks for buzzing along with us today. Until next time, keep those synapses firing, and we'll see you on our next deep dive. Ready to have your minds blown? Today, we're going deep, and I mean deep, into the world of connectomics.
We're talking about a complete, and when I say complete, I mean complete, a complete and densely annotated connectome of the adult fruit fly brain.
Okay, I already know what you're thinking. Who cares about a fly's brain,
Well,
right?
buckle up, because this is way more than just a simple brain map. This is FlyWire.
FlyWire.
FlyWire. It's like having Google Earth, but for a fly's brain. We're talking about seeing every single neuron.
Every neuron. Like, how many are we even talking?
Hold on tight, because we're talking over 139,000 neurons.
139,000. Come on, that's got to be it, right?
But get this, 15 million synapses.
Okay, stop. Synapses. We got to back up for a second. Remind us what a synapse is again.
A synapse is the tiny gap between two neurons where they, kind of talk to each other, transmit signals.
Right. The connections. Okay, so FlyWire is mapping all of these connections in the brain of a fly.
All of them. In 3D, in incredible detail.
But it's still like, why? Why go through all this effort for a fly brain?
This is about way more than just flies. This is about understanding how brains work in general, how they develop, how they learn.
Even a fly's brain.
Especially a fly's brain. It's like a simplified model of more complex brains, even our own.
So by studying something smaller, we can get clues about ourselves.
Exactly. Think of it like trying to understand a jet engine. You wouldn't just look at a jumble of wires and pipes, would you? You'd want a schematic.
Okay, yeah, I see where you're going with this.
FlyWire is like that schematic for the fly brain. It's a game changer.
Okay, that's pretty cool. I'll give you that. But how in the world did they even begin to map all of that?
Yeah.
It's like an impossible amount of data.
It was a massive, massive undertaking, let me tell you. It involved years of work, cutting-edge imaging technology. Oh, and a global community of citizen scientists.
Citizen scientists?
Yeah.
Like regular people helping out.
Thousands of them. They used this special software to help proofread and refine the computer-generated reconstructions of the neurons and the synapses.
Wow, a global team of brain mappers. That's amazing.
It's pretty incredible. And the level of detail they achieved, it's mind-blowing. We're not just talking about location here.
Okay, so what else is there? It can't just be, here's a neuron, there's a synapse.
You're right. The researchers went way beyond that. They developed this really amazing hierarchical annotation system.
Hierarchical.
Yeah, so not only do we know where each neuron and synapse is located, but we also know what it does, its function.
Oh, wow. So they've got it all figured out what each part of the brain is actually doing.
It's like having the instruction manual, finally. And that's not all. They even traced the developmental origins of each neuron. They called these hemilineages.
What now?
Hemilineages. They're basically like family trees for neurons. We can see how they're all related.
Family trees for brain cells. That's next level stuff.
It's incredible. It's like they mapped out a family reunion, but for a fly's entire brain.
Okay, that's a lot of relatives to keep track of. But what's the big deal about these hemilineages, these neuron family trees?
Well, it gets really interesting. By tracing these lineages, the researchers discovered just how ridiculously precise a fly's development is.
Precise? How precise are we talking?
Get this. When they compared the left and right sides of the brain.
You mean like how our brains have two halves too?
Exactly, two hemispheres. So they compared the number of neurons in each hemilineage on each side. You know what the difference was?
I don't know, what, 5%? 10%?
3%. 3% difference. That's it.
Are you serious? 3%? That's insane.
That's nature working with some incredibly tight tolerances, my friend.
Like every fly brain is rolling off the same tiny assembly line, perfectly calibrated down to the last neuron.
It's pretty amazing, but it gets even cooler. Remember how we talked about comparing FlyWire to that earlier partial connectome, the hemibrain?
Yeah, I remember. They wanted to double check their work, right?
Mm-hmm.
Make sure they didn't miss any neurons.
Well, it was more than just that. Comparing the two connectomes allowed them to look for individual variations in brain structure between flies.
Wait, hold on. I thought all fly brains were basically the same. Why would there be variation?
That's a question a lot of neuroscientists are trying to figure out. It turns out that while a lot of cell types are very similar from fly to fly, something we call stereotyped, some cell types show way more variability.
Variability, like what do you mean?
Well, for example, they found that some of the cell types identified in the hemibrain couldn't actually be definitively matched in FlyWire.
Wait, back up. So you're saying some neurons were totally different?
They were different enough that the researchers couldn't say for sure they were the same cell type. And that brings up some really big questions about individuality, even in something as, seemingly simple as a fly brain.
Okay, my mind is officially blown. So we've got this incredibly detailed map of a fly brain. We're finding out that even flies have unique brains. But how do we go from knowing where things are, which neurons connect to which, to understanding what it all means?
Well, that's where the question of connection strength comes in.
Connection strength.
Right. So it's not just about whether or not two neurons are connected. It's also about how strongly they're connected.
Okay, so some connections are more important than others. Is that what you're saying?
Yeah.
How do you even measure something like that in a brain?
Think of it like roads in a city.
Okay.
Some connections are like major highways, multiple lanes, lots of traffic flow. Others are like small, quiet side streets.
Got it. So the number of lanes, that's like the strength of a connection.
You got it. A stronger connection, one with more synapses, means more communication between those two neurons.
Makes sense. So what did they find? Are some of these brain highways consistent across flies while the side streets are a little more, flexible?
Exactly. One of the most interesting findings was that stronger connections, the multi-lane highways, were much more likely to be the same in different flies.
So the core communication pathways in the brain, those are pretty consistent. But maybe the weaker connections, those are the ones that allow for more individual variation.
You got it. It's like the brain has these fundamental highways that are essential for, basic functions, but then it also has these smaller roads that can be rerouted and repaved to create unique individual experiences.
Fascinating. But with all this data, how do they know what they're seeing is real and not just, some kind of error in the data?
That's a really important point. The researchers went to great lengths to differentiate between what we call technical noise and real biological variation.
So how did they do that?
They developed these sophisticated models to account for any imperfections in the imaging data, things like maybe a synapse was missed or a reconstruction wasn't totally complete.
So they basically created like a noise canceling system for brain data.
Something like that. And this incredibly meticulous approach gives them a lot of confidence in the variations that they are seeing.
Wow, that's impressive. Okay, we've talked about how they map the fly brain. We've talked about how they categorize the neurons, how they even figured out which connections matter most. But what can we actually learn from all of this? What's the so what?
Well, that's where a very specific part of the fly brain comes in, the mushroom body.
The mushroom body, okay, what is that? Like the smell center or something?
You got it. It's super important for learning and memory, especially when it comes to, you guessed it, smells.
So like if a fly smells a banana that's past its prime, the mushroom body helps it remember that maybe next time go for a fresher snack.
Exactly. So remember when we were talking about comparing FlyWire to that earlier partial connectome, the hemibrain?
Yeah, they wanted to make sure FlyWire was accurate, right?
Right, but it was more than just that. One of the really interesting things they found was that the FlyWire brain had way more Kenyan cells than the hemibrain.
Kenyan cells, those are, remind me what those are again?
Kenyan cells are like the stars of the mushroom body. They're the neurons that do the heavy lifting when it comes to processing and storing those smell memories.
Okay, right. Kenyan cells, got it. So one fly brain had more cells dedicated to smell and memory. Does that mean like it was a smarter fly?
Well, we can't really measure fly IQ, at least not yet. But what's really interesting is how the FlyWire brain compensated for having all those extra Kenyan cells.
Wait, the brain had to compensate. I thought the brain was like hardwired. How can it just change like that?
Well, here's where it gets really wild. Instead of each Kenyan cell getting input from like a ton of different odor channels.
Odor channels, just a reminder for folks at home, those are kind of like the different scent receptors in our noses, right? Like some are tuned for floral scents, some for, well, you get the idea.
Exactly. So instead of each neuron getting this broad range of smells, the FlyWire brain with all those extra Kenyan cells seemed to like fine-tune things. Each Kenyan cell was getting input from a smally, more specific set of odor channels.
So it's like instead of having a bunch of general conversations, the neurons were having more focused, specialized discussions. They were like experts in their own little odor niches.
Yeah, that's a great way to put it. And this really challenges our understanding of how neural circuits actually adapt to change. Even in a brain as seemingly hardwired as a fly's, there's a remarkable level of flexibility.
That's mind-blowing. So with all this variation between individual flies, how can we really define what a cell type even is? It's like the lines are getting really blurry.
You're hitting on a huge question in neuroscience. And, this study suggests that defining a cell type might require looking beyond just one brain. The researchers are proposing that we need to think about cell types in terms of how similar neurons are to their counterparts in other brains.
So it's about finding consistent patterns across individuals, not just within one individual.
Exactly. And this multi-connectome approach, while it could totally revolutionize how we classify neurons, make it way more accurate, way more robust.
This is really incredible stuff, but I gotta ask, why should anyone besides, a neuroscientist care about what's going on inside a fly's brain?
Well, here's the thing. Even though a fly's brain is obviously way less complex than ours, the basic principles at work are often the same. FlyWire is like this incredible Rosetta Stone for understanding how brains work in general, how neurons connect, how circuits process information, how brains learn and adapt.
So by studying something as small and, well, seemingly simple as a fly, we can actually unlock secrets about our own brains.
You got it. We can start to link specific cell types to specific behaviors, to genes, maybe even to diseases. FlyWire gives us this amazing testing ground for new technologies, new theories that could one day, hopefully, transform how we treat brain disorders.
This has been an awesome deep dive. We started with, the tiniest details of neurons and synapses, and ended up talking about the biggest questions of intelligence itself. Who knew the humble fruit fly was hiding such incredible secrets?
It's a good reminder that there's always something new to discover, even in the most unexpected places.
That's right. And for our listeners, keep exploring, keep asking those big questions, because who knows what other mysteries are out there just waiting to be uncovered.
Okay, ready for this. Today, we're diving into some serious brain science.
Oh, yeah.
Talking about the FlyWire connectome.
Really amazing stuff.
Yeah, you guys sent us this paper, and it's about the first ever complete map of an adult fruit fly brain.
Every single neuron, every connection.
It's incredible.
It really is remarkable, yeah.
It's like, when you think about Google Maps, but imagine it's so detailed that it shows not just every street,
Right.
but every house on the street, every room in the house, every piece of furniture.
Yeah, like every little detail, that's what we're talking about.
That's what this is.
139,255 neurons.
Wow.
15.1 million connections, all meticulously charted.
This was just published in Nature.
Yeah, just last week.
Okay, so it's brand new, hot off the presses. It's packed with fascinating details. But our mission, if we choose to accept it,
I think we should.
is to try to figure out, what are the biggest takeaways?
Yeah, absolutely.
What does this fly brain tell us
Right.
about brains in general?
Right.
Our own brains included.
It's all about, kind of trying to understand those fundamental rules for how brains are built in the first place.
So to get started here, the researchers started by basically making a parts list for the fly brain.
Exactly.
Right.
Yeah, they call it annotation.
Okay.
And they went through and they identified all the different types of neurons, and then they grouped them by their roles.
Okay.
So some of them process sensory information, some control movement, things like that.
And what I thought was really interesting is that they found that there's a surprising amount of consistency between individual flies.
A remarkable degree of consistency, yeah.
Can you expand on that a little?
Yeah, so they found that the left and right hemispheres of the central brain differ by only 0.1%.
Wow.
In the number of neurons.
So
which is really, really tiny.
Each fly is unique.
Right.
But their brains are following this incredibly precise blueprint.
Yeah, it's like there's a blueprint that they're all following very closely.
Like baking a cake, you can use slightly different sized eggs, maybe a little more flour. I don't know.
A little bit of variation.
But you've got the recipe, Yeah. And that's what's really incredible here.
The recipe stays the same.
Is that the instructions for how to put the brain together?
Yeah.
It's incredibly consistent.
Exactly. And I think that speaks to how important it is to get that right.
Right. You don't want a lot of variation in a brain.
You need those neurons to be in the right place.
Even a fly brain.
Exactly.
Right. So speaking of the fly brain,
Yeah.
one thing that really stood out to me was how much of the fly brain is dedicated to vision.
It's huge, yeah.
Over half.
Over half the information that flows into the central brain is coming directly from the visual system.
That's amazing.
It just shows how crucial vision is for a fly to survive.
Well, think about it.
Yeah.
They're navigating, they've got to find food.
They're avoiding getting swatted.
They're avoiding all the fly swatters out there.
Right.
And they're doing all this while relying on these visual cues.
It's all about vision for them.
It's really incredible. And so they're like these tiny little flying acrobats.
That's a great way to put it.
Constantly reacting to the world around them.
Right, and that takes a lot of brain power.
It does.
To process all of that visual information.
And this is amazing. This is really cool.
Okay, what is it?
They found that certain visual neurons, the ones that are sending information from the eyes to the brain,
Yeah.
they actually take different routes
Interesting.
in the left and right hemispheres.
Huh.
So it's kind of like if you have two cars
Okay.
and they're using different paths on a map.
I see, yeah. But they both end up at the same destination.
Exactly.
Well, that's interesting. Which suggests that there's a level of flexibility in brain
I
wiring
really thought.
that we didn't really fully appreciate before.
That's fascinating.
Yeah, so even though we have this precise blueprint,
Right.
there's also room for some variation.
Right.
Yeah, like the brain can adapt
Like different solutions
and still get the job done.
to the same problem.
It's pretty amazing.
It's amazing. Okay, so we've established so far that fly brains are incredibly complex.
Yeah.
They're surprisingly consistent, and they're also really adaptable.
They're impressive little organs.
They really are.
Yeah.
So how does all this help us understand this bigger picture
That's the big question.
of how brains work in general?
Right.
What does this teach us about the fundamental building blocks
Yeah.
of intelligence itself?
That is a great question, and that's actually where the research on cell types comes in.
Okay.
And that is a story for part 2 of our deep
I
dive
can't wait.
into the FlyWire connectome.
Okay, so we're back,
Back again.
and we are ready to go even deeper
Yeah, let's do it.
into this fly brain, deeper into the weeds.
So last time we were talking about how researchers created this detailed parts list
Right, like a parts list for the brain.
of neurons.
Yeah.
And now we're going to talk about how all those parts fit together
Yeah.
to form different cell types.
This is where it gets really cool.
Okay,
This is the really interesting part.
let's do it.
So the paper you sent
Yeah,
compares this new complete FlyWire connectome
right.
to the older partial map called the hemibrain.
Yeah, the hemibrain.
And this was a big deal
Huge at the time.
when it came out.
Yeah.
But this new map
It's like night and day.
just blows it out of the water
Right.
in terms of completeness.
And when they compared the two,
Yeah.
what did they find?
So they found something pretty unexpected.
Which is that while a lot of the cell types matched up perfectly between the two,
Okay.
a surprising number did not.
Really?
Yeah, it's kind of like,
So some of the pieces.
yeah, like if you had a Lego
Look
set
at the instructions.
and some of the pieces just, no, so they don't fit.
What does that mean?
No.
Does that mean that the old map was wrong?
Well, not necessarily.
Okay.
What it really points to is this question of, like,
Yeah.
how we actually define cell types in the brain.
Right. Because, I mean...
Because you would think,
Yeah.
you know, a neuron is a neuron.
Right.
But it turns out it's not quite that simple.
So traditionally, how have we been defining them?
So we've been defining them based on their shape
Okay.
and their location.
So kind of how it looks.
Like if you were trying to identify trees, you look at their leaves and their bark.
And the branches and everything?
But this research suggests that, you know what,
Yeah.
we need to consider their connections too.
So not just what it looks like.
Right, because it's not just about
Yeah.
what a neuron looks like in isolation.
Like who it's talking to.
It's about,
It's social network in the brain.
yeah, exactly. It's like, who is it connected to?
Yeah.
What signals is it sending and receiving?
That makes sense.
And so they came up with this new definition,
Okay.
which I really like.
I'm ready for it.
They said, a cell type is a group of neurons that are more similar to each other across different brains than they are to other neurons in the same brain. So it's about finding...
So even though the brains themselves are different,
Yeah, exactly.
the cell types are consistent
The cell types are consistent.
between them.
Yeah, it's about finding those consistent patterns
Interesting.
of connectivity, the neural circuits that repeat from fly to fly,
Okay, so kind of like how, like,
you know.
houses can look different on the outside, but underneath they've got the same, like,
They've got the same basic blueprint.
Plumbing and electrical, that kind of thing.
The arrangement of the rooms. Yeah.
Yeah.
The plumbing, the electrical wiring.
It's like finding the brains, like universal language.
The universal language of the brain. And this has huge implications.
Yeah, like what?
Well, for one thing,
okay,
it could completely change how we build computer models of the brain.
Because if we can find those...
Because right now...
deleting patterns.
We're trying to, like, model every single neuron, and it's too much.
It simplifies it.
But if we can identify those consistent building blocks...
makes it a lot easier.
Those repeating patterns... then it becomes a lot easier to create these models.
So it's like we were talking about with the blueprint.
Exactly.
Right. Instead of trying to, like...
instead of having to invent...
custom make every single
piece,
right?
thread.
We have the Lego bricks now.
We can just, like, say, okay.
Yeah, we've got the instructions.
We need 4 walls made of this.
And we know what they should look like.
And then we need, 5 windows of this design.
Exactly.
So it just becomes a much more efficient process.
Which is what the brain is all about, right?
The brain is all about efficiency.
Efficiency. Okay, so we've gone really deep here.
Yeah, we're deep in it now.
Into this fly brain.
We're practically flies ourselves at this point.
Pretty much. And we've explored all this intricate wiring and these surprising consistencies.
It's amazing stuff.
But I gotta ask,
yeah,
if even a tiny fly brain
right.
is this complex, how does all that information even fit in their DNA?
It's a great question.
It's like trying to fit.
It's a huge question.
A map of the whole world.
It's like one of the big mysteries.
On a single piece of paper.
In neuroscience.
Right.
It's called the genomic bottleneck.
Okay.
Which is
So the fly genome is actually pretty small.
Yeah, relatively speaking.
Yeah.
It's small.
How do they do it?
Yeah, it seems impossible.
Is it?
That it could actually, like, contain the instructions for all of that complexity.
Right.
For every single connection.
Exactly.
And, we don't have a complete answer to that.
Okay. But this research suggests...
Yeah, what this research suggests is that this cell typing that we've been talking about.
Okay.
The way that neurons are organized into these repeating patterns.
Right. Okay.
That might be a big part of the answer.
Okay, I'm listening.
So think about it like this.
Okay.
Imagine you're trying to, like, write a set of instructions for building a house.
Okay.
You wouldn't, like, list out the exact location of every single brick, would you?
That would be, I mean, you could, a very long instruction manual.
But it would be, like, incredibly inefficient.
Yeah, no one would want to use that.
And really, really long.
Yeah, exactly.
So instead, you would group things together.
Okay.
You'd say, okay, build 4 walls using this type of brick.
Makes sense.
Install 5 windows of this design,
Right.
So it's like you're creating these higher level instructions.
Yeah.
And I think cell typing might be the brain's way of doing something very similar.
So instead of, like, dictating every single connection, it's giving these, like, broader principles.
Yeah, these kind of, like...
and then letting development...
principles.
I have to figure out the rest.
Exactly.
So it's like the genome is what, like the blueprint?
Yeah, I like that analogy.
And then development is...
like the genome provides the blueprint, the basic melody.
Okay.
And then development is like the improvisation.
I like that. Okay, so that's starting to make a little more sense.
Yeah.
But then that brings up another thought.
Okay.
Like, if that's how fly brains work, what does that tell us about our own brains?
Right.
Because our brains are...
I mean, think about it.
Exponentially more complex.
Our brains are, like, orders of magnitude more complex than a fly brain.
But could these same principles
and yet
be at play?
Yeah, that's the question, right?
Right.
And I think it's definitely a possibility.
So it could be more similar.
Because if it works for the fly, it might work for other brains too.
And we thought.
Right. And if that's true, then that means that by studying these, quote unquote, simple creatures, like flies, by mapping their brains, by trying to understand how their neural circuits work.
We're actually gaining insights into our own brains.
Yeah, we're gaining insights into the fundamental principles of intelligence itself.
Wow. So this research isn't just about flies. It's about, like...
It's not just about the flies.
What does it mean?
It's about us.
To be intelligent.
It's about understanding.
That's amazing.
The very fabric of thought, of consciousness, of what makes us who we are.
Well, we started with a map of a fly brain.
That's the beauty of science, right?
And now we're here.
You never know where it's going to take you.
It's incredible.
You start with a simple question.
Yeah, it's like we're contemplating the very nature of intelligence.
And it leads you down these rabbit holes.
Yeah.
It opens your mind to new possibilities.
And that's the beauty of science.
Exactly.
That's what we love about it.
It's about the journey.
So what if the key to all of this, to unlocking, like, the mysteries of our own minds,
What if?
lies not in looking out
Yeah.
at the vastness of the universe,
At the stars.
but in looking at the intricate world
At the smallest creatures.
of a fly's brain?
It makes you think.
It really does. It really does. And on that note,
Yeah.
we'll leave you with that thought.
Go out there and look at some flies.